Hi, I'm Doug Noll from University of Michigan, and my project that I'm talking about today is high SNR functional MRI using oscillating steady-state fMRI. Let me give you the overall goals of the project. We would like to boost the signal-to-noise ratio, or SNR, for high-resolution fMRI. We hope that our methods can be applied to standard MRI instrumentation, and the long-term goal, again, is high-resolution fMRI, so we would like to enable columnar or layer-specific fMRI without the need for purchasing a higher field scanner. Let me summarize the oscillating steady-state, or OSSI, fMRI first. So OSSI is a completely new method for fMRI. It provides T2 star weighted or bold light contrast that most of us have gotten used to for fMRI studies. And it has a boosted SNR on the order of twice as much SNR as standard GRE methods. And if it was that simple, we'd be done, of course. But the method does have a fairly complicated image acquisition and reconstruction process. It is also fairly sensitive to physiological noise and possibly also to head motion as well. And that's also something we work on. And lastly, it's slower. So that is, the volume TR is lower than standard GRE, and we need to address all of these issues in the grant. Let me start, first of all, with some of the basics of signal-to-noise ratio in functional MRI. So this formula that we have here, that is the SNR is proportional to the volume, that's the voxel volume of the imaging element, the field strength, and then some other parameter related to the total acquisition time. This is sort of just a general formalism for signal-to-noise ratio in MRI. Let me focus on that last one, the total acquisition time. So in general, that's related to kind of the length of the readout window and the number of averages. But in a specific case that we're talking about here, which is in gradient echo fMRI, when we're imaging at the Ernst angle, basically, that more or less ends up being proportional to the number of averages, or if you will, the number of time frames times the TR, right? And so for shorter TRs, we have smaller flip angles, and less signal for each of them, but then we have proportionally larger numbers of averages, right? And so it turns out that at the Ernst angle, this ends up being nearly a constant for most GRE imaging. Of notice, some things that are not included in that expression, which is the total imaging volume, the number of slices, the field of view. Turns out those things don't really matter from the standpoint of the signal-to-noise ratio. Now, why is this important? Well, if again you take a look at this, suppose we want to do high-resolution imaging. So, for example, suppose we wanted to have that voxel volume by a factor 2. Have the voxel dimension by a factor 2. That means we reduce the voxel volume by a factor of 8. Well, if we wanted to correct for that by increasing field strength, well, you know, maybe a factor of 8 in field strength, which would send us up to very high fields. And maybe it's a little bit less than that because the Bowl effect gets larger as well. We could make up for that by increasing the total acquisition time, but that's very expensive because it goes as a square root of the acquisition time. So that would actually make the exam extremely long and essentially infeasibly long. So if there's other ways to improve SNR, all the better. So now this OSSI technique is based on kind of an oscillation of results in an oscillation of the MRI signal, and that comes from a quadratic phase sequencing. So quadratic phase sequencing is common in MRI. It's often used in spoiling, but if we have balance gradients, which is what we're proposing here, then we actually get an oscillating signal that goes with the periodic nature of this quadratic phase pattern. And the details of this are not important, but note that we have a linear progression of the phase differences from RF pulse to RF pulse. And if we make that kind of a nice number, it repeats periodically. So here's an example of that. So here's a phantom that we have a quadratic phase sequence that repeats 10 times. And so here's the first sequence of 10 images, the next sequence of 10 images. And you can see that there's an oscillating pattern that repeats every 10 images, right? Okay, if we add spoiling gradients, this essentially becomes spoil imaging. We have a nice steady signal, which you'd think would be good, except the signal strength is a lot lower. And so our average signal here is a lot higher than the signal here. Okay. So how do we use this for fMRI? Well, we need to combine these oscillating signals, as you see here, and we can combine them numerous different ways, and that's one of the things we're looking at. But so, for example, you can combine them with a 2 norm across these and produce an image here. And these images then can be used for, say, for example, for fMRI. Now, it turns out, and I'm not going to go into the arguments why, these images end up being effectively T2 star weighted or bold contrast weighted and can be used for fMRI, just like GRE. Okay, and so does it work? Yeah, of course it works. And so this was some preliminary data that we acquired many years ago, where basically we took our combined images, the oscillating steady state or OSSI images, and those are shown here in the background, and we can see the activation areas shown here. We can see the fMRI time course, the TSNR maps. You can see that we have an improvement relative to GRE imaging. Okay, and so as you can see, it's quite promising. It's a quite promising method. We ran a preliminary study. This came out actually came out in 2019 as a preprint and is in press in 2020. We did a more established study comparing oscillating steady state to GRE, and you can see we have boosted SNR, and that's and kind of in more quantitative numbers here. We had about 1.5 times the number of active voxels, and we had 1.74 times the SNR for this particular study. Okay. So let's talk about the aims of the grant. So the first aim is to analyze the spin physics of oscillating steady state fMRI. The second aim is to optimize the acquisition and reconstruction or improve the speed and SNR of the imaging. There we have a couple of targets that we're shooting for. So in terms of temporal resolution, we're shooting for a volume TR of 2 to 3 seconds. So this is not going to be as fast as, say, simultaneous multi-slice imaging. And we have a target spatial resolution, and we have a couple different studies proposed, but one of the goals is to get to 0.8 millimeter isotropic spatial resolution in our final version of this. Aim 3 is to try to address some of these artifacts. So for example, the physiological noise that I talked about before is worse in oscillating steady state as compared to GRE. And so we need to address that. And we're also going to look at motion artifact. Lastly, in the final aim, we're going to looking at evaluating the method. So we have we have an activation phantom and a resolution phantom. So we're going to be examining some of the imaging properties. And then lastly, we're going to be doing comparison to leading edge methods. So for example, simultaneous multi-slice, GRE, EPI, kind of effectively the state of the art for fMRI at 3T. We're going to be doing a comparison with that. So let me talk about some of the progress we've made along the way. So for example, in terms of speeding the acquisition under aim 2, we've looked at a number of different sparse sampling approaches. On the left, we have a kind of a stack of variable density spirals, and these are fairly sparse spirals. So for example, you can see this is a total matrix size of about 160, but you can see we only have a few turns of the spiral. So it's a rather sparsely sampled spiral, and it's stacked in 3 dimensions in this way. And then these spirals end up rotating with golden angle type rotations from run to run. On the right is another method we've looked at that we call radial echovolume imaging, or REVI. This also kind of traverses both KZ as well as KX and KY with a similar spatial resolution, about 160 matrix size. And this whole structure here effectively rotates, again, with sort of golden angle type rotations. And so these are examples of some of the acquisition methods that we've been looking at. In terms of the image reconstruction, what we really are trying to do is take advantage of some of the reproducible nature of the oscillation. So as you can see from these 2 lines, it's very reproducible from run to run. And that means we might be able to take advantage of some sort of structure within the data. And kinds of things we've been looking at is low-rank tensors. So this includes 2 time dimensions, both this fast time and the slow time, as well as spatial dimensions. So for example, 3D or 4D, or even 5D if you count all 3 spatial dimensions. And so we have fairly large tensors, and it becomes a low-rank tensor kind of constraint. I'll talk a little bit more about this in a second. We have dictionary models, so we can model this sort of variation of the signal via dictionaries. and use that in image reconstruction, kind of way to constrain the reconstruction. And we've been also looking at unrolled neural networks as regularizers, again, trying to learn this structure via neural networks. Here's an example of the low-rank tensor models. So for example, if we stack the data, this is oscillating variation in this direction and also slow variation in this direction, and we kind of stack it up, this oscillatory dimension, what we call fast time, slow time, and spatial dimensions, and we end up with a structure. I've shown it as 3D, but it can also be 4D or 5D. And then we have various unfoldings, and we can look at and enforce kind of low-rank constraints along those various dimensions. Well, that works pretty well, and this is an example of that. So here we have a fairly high resolution acquisition, 1.3 millimeter spatial resolution with 150 millisecond temporal resolutions. This is just one slice, however. And you can kind of see that we have, with the tensor reconstruction, we have very nice SNR and good activation maps. If we just did sort of a standard sense type reconstruction, conjugate gradient sense reconstruction, you can see you have a much noisier case here, and it doesn't work nearly as well. But this tensor reconstruction allows us to exploit and extract higher SNR, higher temporal resolution, higher spatial resolution from our acquisition. And this is an example of how we're using this method. Our timetable, well, again, we're right in the middle of our project right now. We're almost exactly halfway through. We've made a lot of progress on the image acquisition and reconstruction. Our acquisition side of it is going to be kind of hopefully winding down, but we can still make developments in the image reconstruction side and apply those to data that we collect later on, as long as the acquisition kind of stays more or less fixed. Our spin physics work is mostly done, doing a little bit of work on modeling of motion and flow, but that's mostly done at this point. And then our methods for trying to correct for motion and flow are going to continue out into year 4 as well. We've been doing phantom validations along the way, and then starting next year, we're really going to start to get more into the human subjects validations. Let me make a couple comments about dissemination. So obviously the first point of dissemination for work like this is publications. We'd like to publish. We have been publishing regularly and will continue to do so as a way of kind of getting our ideas out into the field. The image reconstruction code, we have a long history in our group, particularly with my collaborator on this project, Jeff Fessler, who has the Michigan Image Reconstruction Toolbox, which is online. We have a long history of posting code there, and we'll continue to do so with this project. In terms of sharing and disseminating acquisition methods, we haven't made any firm commitments to do this, but we're looking at distributing this via some platform or vendor independent languages. So for example, there's a project with one of my colleagues on this project, John Nielsen, with Maxim Zaitsev at University of Vienna, using languages that we call TOPI or PulseSeq, which are kind of platform independent languages, and we have a translator that goes between these. And so whatever we do on GE, we can set up almost to run almost exactly the same on the Siemens scanner. And so we're hoping that we might be able to share things this way. So in summary, our OSCI method is a new imaging method for fMRI. It produces high SNR, at least with respect to thermal noise, making it suitable for high resolution fMRI mapping. It uses existing instrumentation. We've done nothing special in terms of the hardware, it just uses it. But it does require some sophisticated model-based image reconstructions to make it fast enough to be useful. And lastly, because it is sensitive to physiological noise and motion, we're spending a lot of effort to try to make that better. I'd like to acknowledge my collaborators. Jeff Fessler, as I mentioned, is an image reconstruction guru. John Nielsen is working on a lot of the coding, and my collaborators are more on the neuroscience side, Scott Peltier and David Braun. I have a group of students who've done a lot of work on this, and their names are listed. And again, thanks to the NIBIB and NINDS for the support via the BRAIN Initiative. Thanks.